is a trustee of the Ciliopathy Alliance, and he works in the University of Edinburgh. He's an academic ophthalmologist with a clinical and research interest in the inherited retinal dystrophies, a group of untreated genetic conditions caused by a single faulty gene. And they are the leading cause of visual loss in children and working adults. He runs the Inherited Retinal Dystrophies Service in NHS Lothian, which is up in Scotland for our overseas guests. And his research is in interventional clinical trials, including gene therapy and exon skipping therapy, as well as natural history studies, amongst others. His research lab uses animal and stem cell models to better understand how inherited retinal dystrophies develop in the eyes and define the pathways disrupted in disease that eventually lead to retinal degeneration, with the ultimate aim of identifying new treatments. So thank you, Roly, for coming today, and we really look forward to hearing what you've got to tell us. And did I get it all right, or did I make it up?
Spot on.
Thank you.
So I am just going to share my screen. So the first question, Fiona, before you mute yourself, is can you see my screen, and is it full present? Is it on slideshow?
I can, and it all looks perfect. Yeah, thank you.
Great, fabulous. Okay, thanks very much for the introduction, and thank you for the invite to speak, and thank you to everybody for coming along. I think I've got about half an hour, and I am going to divide my talk, if I can advance my slides, initially just to speak a little bit about the approaches to treating inherited retinal dystrophies that are close to or that are in clinic at the minute. And that, of course, I'm sure, as everyone would guess, would involve gene therapy, which I'll spend a little time talking about. I'll also speak a little bit about the non-gene therapy approaches to clinical trial. That are in clinical trial for treating IRDs, and then I'll speak a little bit about my lab's work, and I'll just speak about one story that we just recently have published. I'm aware that there are no doubt some folk on the call with an inherited retinal dystrophy, and it always throws up the quandary as to whether you use slides or not, because some of the folk here might not be able to see my slides particularly well, what I'm going to be showing. I've decided to show some slides today, but I will try to explain them as best I can. It just gives me a little bit of a script to work off. So yeah, this is obviously a Ciliopathy Alliance meeting, and ciliopathies, as Fiona has alluded to, can cause retinal degeneration as well as lots of other multi-system disorders, and that's why I'm going to speak to you about the effect that ciliopathies can have on the eye and on vision. I'm going to start the talk today by presenting a photo here of a small single-celled flagellate organism called Euglena. And the reason I show this is because 650 million years ago, when we were all single-celled organisms, we were all floating around in sort of a primordial soup, not being able to interact at all with the world around us. The Euglena underwent a mutation in its DNA, as we are all constantly doing in all our cells. And as a result of that mutation, it started making a protein which was sensitive to light. Now, this meant that it could start to appreciate the world around it, but because it was single-celled, it couldn't actually interact with it, or it couldn't process any of this information, and therefore life on Earth started having to build central processing units behind this light-sensing protein. We call these central processing units the brain, and it's just a point just to show that the contrary to what a neurologist might tell you, the brain is actually an extension of the eye, not the other way around, and the eye did come first. You fast forward 650 million years and we have evolved massively, and obviously humans now have a very much more complex way of interpreting the world around us. I'm showing a very rudimentary cartoon of a human eye, but at the back of the human eye you have a three-layered structure called the retina, and it is absolutely beautiful in its simplicity, because this set of three layers of cells are all you need to be able to turn electromagnetic radiation into an impulse which can go to your brain and therefore allows you to see. And there's an argument, given that we are now addicted to and pretty much attached to our smartphones. There is an argument that we're possibly using our sight more than ever. So whether or not food porn is your thing, we are really using our eyes because of our our increasing addiction and reliance on smart phone technology. And the site of the initiation of the visual cascade is the photoreceptor. At the back of your eye, you've got about 120 million each of these cells in each eye. And whilst it is traditionally, the photoreceptor cell is divided into an inner segment and an outer segment, divided up by what we call a connecting cilium, in actual fact, the entire outer segment is a highly modified primary cilium. And cilia, as most of you here know, are the sensing organelles in our body. They stick out like an antennae from the vast majority of every single cell in our body. And as a result of these cilia, each of our cells can sense the environment that they're in and respond accordingly. Now, cilia signal largely through a protein called a G protein coupled receptor, which is concentrated in the ciliary membrane. And it was shown about 7 or 8 years ago that cilia regulate the amount of G protein coupled receptors on their membrane by sizing off and by getting rid of excess G protein coupled receptors by shedding vesicles from the tip of the cilia. But in contrast to this, the photoreceptor has evolved to produce this huge expanse of ciliary membrane, which folds back and forth in itself hundreds of times to form hundreds of disc-like processes, which are stuffed full of the light-sensing G protein coupled receptor, and we call that rhodopsin. And so the photoreceptor has hundreds of discs which are formed from this expansive membrane. And in this extraordinary feat of biology, this entire outer segment is completely renewed every week in our photoreceptor. And that requires a massive amount of trafficking of protein and a massive amount of deformation of the ciliary membrane. And whenever that goes wrong because of human mutations, patients develop an inherited retinal dystrophy. And as Fiona says, these IRDs, as we call them, are the leading cause of visual loss in children and working adults. And I'm showing 2 pictures here. One is the back of a healthy eye, and one is the back of a patient with a disease called retinitis pigmentosa, which is the commonest IRD. And it's really a bit of an umbrella term, an IRD or retinitis pigmentosa, because although the patients, whenever you look at the back of their eyes, they have this bone spicule formation, this sort of blackness emerging in the surrounding of the eye, hence the name pigmentosa. They also have narrow blood vessels and they have very pale optic nerves. But really that's just an umbrella description, because over 280 genes have now been identified, mutations in which can lead to a retinal dystrophy. And until very recently, none of these were treatable. And so if you want to divide them up into 280 diseases, which really that's what they are, none of these were treatable. But the last 20-30 years has seen the advent and the development of gene therapy for human conditions. And what gene therapy consists of is you have the vast majority of monogenic diseases, and when I say monogenic I mean diseases which are caused by a mutation in a single gene. The vast majority of those mutations result in no protein being produced by your body. So DNA makes RNA and RNA makes protein, and the vast majority of genetic conditions where you have misspelling of your DNA, it means that you don't make any of the protein that gene is supposed to make. And so what scientists can now do is they can take human DNA and they can package it into a virus. Now, viruses are brilliant at invading cells. You know, we all get colds and flus every winter, and that is because the viruses are so good at transducing, at getting into human cells. And so in theory, if you can package a human gene into a virus, you can then put it in the body where it is required, where disease is developing, and in the hope that virus will infect the cells which are not functioning properly and start to make the healthy protein that is missing in that cell. And so we have developed this in the ophthalmology world, and we can design gene therapies which we can inject into the back of the eye underneath the retina. And in theory, these viruses can then infect the light sensing cells, infect these photoreceptors, in the hope that the normal human protein can be made and can hopefully rescue vision. And I'm showing you a picture here of a Briard dog called Lancelot. One of the rarest, a rarer form of inherited retinal dystrophy is caused by a mutation in a gene called RPE65. And the Briard dogs are obviously very inbred because we all like a pedigree dog. And as a result, inbred pedigree dogs harbour genetic disease. And the Briard dog has a mutation in the RPE65 gene, so they go blind just like humans go blind. And scientists, going on for 20 years ago now, in Philadelphia, took Briard dogs who had mutations in RPE65, so they had the condition called Leber's congenital amaurosis. They went blind, and they developed a gene therapy where they made the human RPE65 gene, they put it into a virus, and they put it into the back of Lancelot's eye, and Lancelot's vision improved hugely. And this was sort of proof in principle that in a large mammal you could rescue vision by delivering gene therapy to the back of the eye. Lancelot then became pretty much a household name. He met, I think, 2 US presidents. Apparently he was on the Oprah Winfrey couch, although I can never find footage of that online to give him presentations. But he went on a sort of promotional trip, and he raised a lot of money that went into the company that designed this gene therapy trial, this gene therapy product. And as a result, a human trial was born, and that showed that this human gene could also rescue vision in humans. And as a result, we now have our first NICE recommended gene therapy for delivery on the NHS for free to patients who have mutations in their RPE65 gene. And so it's been an amazing journey for gene therapy, and because the eye is a relatively isolated organ, it tends to be the leader in advanced therapeutic trials. And so we now have a licensed gene therapy product for one very rare form of eye disease. It is a bit of a cautionary tale. We now can give this, and so worldwide, well over 100 patients have been treated now with, well over 200 patients have now been treated with Luxturna, which is the approved gene therapy. But what we are starting to appreciate now with long-term data in the real-world setting is that patients can develop an inflammatory reaction at the back of the eye due to this gene therapy. It's not thought that it's due to the surgery itself, because the inflammatory reaction results in loss of your light sensing cells, and this can occur out with the surgical site. So we think that there is some form of inflammation being driven by the gene therapy product. It's only in a small set of people, but in those small sets of people, you get atrophy, you get wasting at the back of the eye, and so you get progression of sight loss. So although it is definitely a huge hope when we finally have a treatment for retinal dystrophies, it does come with somewhat of a note of caution. Gene therapies now, therefore, now that we've shown a proof of principle that with a very rare form you can target retinal dystrophies with gene therapies, we now have a slew of gene therapy trials. We ourselves in Edinburgh are taking part in one. It's for a much more common gene called RPGR, which causes retinitis pigmentosa. And I'm showing a video here of the type of surgery which you need to do to deliver gene therapy to the back of the eye. So at the back of your retina, a little retinotomy is made, a little hole in the retina, and you then inject the gene therapy underneath this retina, which causes a retinal detachment, which then seals itself and reattaches, and you then have virus underneath the eye, underneath your retina, which can hopefully infect your photoreceptors. So we're taking part in this gene therapy trial. We've treated a couple of patients, and it seems to be a very low risk procedure in the short term. There's not a lot of inflammation in the eye, and the patients seem to recover quite quickly. And we'll know over the next 18 months or so whether this gene therapy product helps rescue vision in X-linked retinitis pigmentosa due to mutation in an RPGR. There are several other gene therapy trials in trial at the minute. One is for a mutation in the gene GUCY2D, which causes, again, a severe form of sight loss, Leber's congenital amaurosis, where patients are born with very severe sight loss. Atsena Therapeutics from Philadelphia are carrying out the trial, and results from the phase 1/2 trial at a meeting I was at last month suggested that the treatment is safe, and there is no worsening of the vision whenever patients receive the gene therapy trial. And there appear to be improvements in some of the primary endpoints, including patients' ability to navigate around a maze at low light levels, an improvement in their visual function questionnaires. So whenever patients are themselves asked, how do you feel this has affected your vision? And also something called FST, which I'll speak about shortly. And the amazing thing to note in this trial was that there was a 76-year-old person was treated who appeared to have improved sensitivity in the retina. Another disease which has been undergoing a clinical trial, a gene therapy trial, is Batten's disease, which is due to a mutation in CLN2 gene. Mutations in this gene leads to a deficiency in a protein called tripeptidyl peptidase, and it causes a really severe systemic disease where children develop rapid onset dementia and seizures and visual loss. Patients are dead by the age of 10. It really is a very, very severe disease. But we now know that if you put a reservoir in the brain to replace the missing protein, it halts the neurodegenerative decline, and therefore patients are not dying of the dementia and seizures and the brain degeneration. And therefore the question is, could you therefore rescue vision in the eye? And so a gene therapy product was designed which essentially infects all cells at the back of the eye, and leads to sort of like a biofactory making a replacement of this TPP1 gene. And Rob Henderson at Grey Norman Street has treated patients in a phase 1/2 trial, which is a safety trial. But what he's shown is that the gene therapy slows the thinning of the retina. However, what they have also discovered in a sort of a trial going on at the same time is that if you just replace the protein itself with injections into the vitreous in the eye, much like you would get for macular degeneration, that's enough to show visual improvement. So in fact, this gene therapy pipeline is being stopped because a slightly less invasive treatment is shown to be effective. So those are a couple of examples of gene therapy trials, and which are currently underway. But there are other approaches to treating inherited retinal dystrophies. One is to treat RP caused by mutations in a gene called PRPF31, which is a slightly more common cause of RP. And John Grigg in Sydney has led the platypus study, which is targeted the disease by injecting into the vitreous. Again, this is injecting into the vitreous is a much more simple, non-invasive way of treating somebody rather than the subretinal delivery which is required for gene therapy. So they have this company have designed a drug that blocks the blocker of PRPF31 and therefore increases the amount of PRPF31 in the eye. And by delivering this treatment into the vitreous, he has shown in his phase 1 trial, in his safety trial, that the treatment is safe, and that there's a possibility that microperimetry, which is a way of assessing your central visual field, shows a little bit of improvement. So these patients would need an injection every 3 months if this was shown to be effective in a proper phase 3 study. But it just shows that there's an alternative way of targeting an IRD. One other way of potentially treating retinal dystrophies is with optogenetic therapy. So with optogenetic therapy, what patient, or what you can do is, whenever your photoreceptors are completely degenerate, gene therapy won't work because there are no photoreceptors to receive the gene. So what people are trying to do in that situation is to design a gene therapy which would transfect the bipolar cells, which are the next order of neurons at the back of the eye, which are involved in the visual impulse to deliver vision to the back of the eye. And the gene in question would make a light sensing protein, which would then get into the bipolar cells, and so the bipolar cells would then become light sensitive rather than the photoreceptors, which have all died off. And so, again, a safety trial has been carried out for this approach, and it was presented again at this meeting last month that we were at, and it showed that they treated 18 patients whose vision was counting fingers or worse. So these are patients who have severe visual loss, and they showed that they assessed visual changes by looking at the patient's ability to see objects on a computer, and almost half of the patients had some degree of visual improvement. So this is very, very preliminary data. Like I say, this was just a safety trial, but it just shows that there's a potential approach to treating an IRD when patients have already lost their light sensing cells. And the tremendous thing about this treatment is that it's what we call mutation agnostic. And what I mean by that is, no matter the mutation you have that has caused your sight loss, this treatment would be you would be amenable to this treatment. So whereas gene therapies have to target every single gene one at a time, this one could be delivered to a patient regardless of the underlying mutation. The last treatment I'm going to speak about that's in clinical trial is something called exon skipping therapy. And this is there's a clinical trial about to get up and running to treat Usher syndrome, which we'll hear about later on. So Usher syndrome, caused by a mutation in a gene called USH2A, causes patients to be born deaf and go blind because of retinitis pigmentosa. And the majority, or rather a lot of the mutations that occur in USH2A occur in a part of the gene called exon 13. So genes, as I said, make RNA, which makes protein, and genes are made up of exons, which are split up on the genome by introns. And whenever you make your RNA, you remove all the introns and put all the exons together, which then allows the full-length protein to be made. And a company, or rather a research group in the Netherlands, have shown that if you remove exon 13 from USH2A, you still get a functional protein. And because patients have the majority of patients have their mutations in exon 13, the question is, could you make a protein missing that exon 13, which would allow rescue of vision? And so this exon skipping therapy works by binding to the start of exon 13, which means that the light sensing cells make USH2A without that protein, and therefore you don't get the mutation in the protein, and therefore you get a protein being made. And they have shown in their animal models that this can rescue vision. And so a company called Sepal Bio are beginning a safety trial for this therapy, hopefully before the end of the year. It will start, and if not next year, and Edinburgh is hoping to be a trial site for that treatment, for that trial. Before I go on to a little bit of work from our lab, I just want to very briefly mention the endpoints for gene therapy clinical trials. So because of the big diabetic studies that took place 40 years ago, which showed that laser therapy at the back of the eye could improve vision in patients with diabetic eye disease, those treatments were able to show quite a significant improvement in vision. And as a result, the FDA, the Food and Drug Administration in America, have always required any treatment for a blinding condition to show this a similar amount of visual improvement. Now, gene therapies are just at the minute not going to deliver that sort of visual improvement, and therefore people have been working very hard to develop novel endpoints for clinical trials where they can show improvement for patients that doesn't require this huge improvement in visual acuity. And so things that they have developed are maze tests, where they have in hangars they've built huge mazes, where patients can navigate around them at varying light levels, and it's now been recognised by the FDA that if you can show improvement in a patient's ability to navigate around this maze at a lower light level than before they got the treatment, then that is a realistic primary endpoint. Microperimetry is another one, which is microperimetry is a way of assessing your central visual field, and that is sort of increasingly being used as a primary endpoint for clinical trials. But actually, the improvement, one of the primary endpoints that led to the Luxturna gene therapy trial being approved and being available on the NHS is something called FST, which is just a full field threshold sensitivity. So essentially, you have bright light is shone at a patient, and if they can see if they can detect a lower flash of light after the surgery than before it, then that is seen as a legitimate endpoint to show improvement in vision. So just to summarise this part of the talk, as far as approaches to treating IRDs that are in clinical trial, we've got one gene therapy available in the NHS. We've got several more gene therapies in clinical trial in the UK and beyond. There are other therapeutic approaches being explored, and realistic endpoints for these trials are still an issue that we need to resolve if we're going to demonstrate proper, if we're going to get gene therapies and other approaches approved for IRDs. So I'm going to very quickly just talk about the work we do in Edinburgh. I work at the MRC Human Genetics Unit at the Institute of Genetics and Cancer at the University of Edinburgh. And what we're trying to do is try to understand human eye disease across scale. So from nanometers right up to, to humans. And so we do this with several approaches. We're looking at DNA. We're trying to improve the diagnosis rate of patients with genetic eye disease. We're using large data sets from single cell and bulk RNA sequencing and proteomics to try to better understand disease pathways that are disrupted in IRDs. And we're then using a sort of advanced imaging technology to try to really get at the mechanism of disease, why these mutations are causing people to go blind. And then at the slightly bigger scale, we have animal models with which we can investigate these diseases. So I'm going to go back to what I said at the start about how the photoreceptor cilia is highly specialized, and it has this huge big outer segment which is stuffed full of our light sensing pigment, and how this constant turnover of this outer segment is required for vision. And the gene I'm going to speak about today is RPGR, which is a gene which causes about 90% of X-linked retinitis pigmentosa. So it causes about 15% of all RP. So it's one of the major causes of RP. And like I say, we're doing a gene therapy trial for RPGR, but at present there is no effective treatment for it. Now, the reason why it's important in the context of outer segment turnover is because I'm showing a picture here of an OCT, which is an optical coherence tomography of the back of the eye, and I'm showing a healthy eye, and at the back of the eye, because of this OCT, you can delineate the outer segment, so you can see a white band on the image, which is the outer segments of the photoreceptors. And in patients with RPGR mutations, they lose those outer segments. So patients with RPGR appear to lose their outer segments on imaging. Now, RPGR is thought to localize to the base of the outer segment where these discs are made, where these outer segments are continually renewed. And the work when we started our work, we were using stem cell technology to model RPGR disease. So what we did was we took stem cells, or rather we made stem cells from patients with RPGR mutations who had retinitis pigmentosa. And we did that using a technology which won the Nobel Prize about 15 years ago, which showed that if you took an adult cell, in this case a skin cell, and you overexpressed 4 genes, you could turn the adult cell back into a stem cell. Now, stem cells are called stem cells because they have the ability to self-renew. So whenever you make them, they're never ending. And they also have the ability to differentiate into any cell in the body. And in this case, of course, we were interested in light sensing cells. And I'm showing a video here of some work done by the lab of Yoshiki Sasai in Japan. And what Yoshiki Sasai showed was that if you take these stem cells and pattern them towards a ventral brain fate, so if you push them towards being frontal brain cells, they have the inherent ability to organize themselves and fold in themselves and form little eye cups in a dish. And so we took patients with RPGR mutations, we made stem cells, and we grew these little eyes in a dish. So whenever you grow them for about 50 days, they start to sort of organize themselves and fold in themselves. And if you grow them for about 150 days, they mature, and you have these tiny little eye cups, which whenever you section through them, you can see a lovely layer of stratified light sensing cells, which you can then use to model the disease. Because what you can do is you can take patients with RPGR mutations, you can take their unaffected relatives, and you can compare whenever you grow these mini eyes in a dish, you can compare one to the other. And what we saw was that when the patient derived eye cups, the little mini eyes, whenever you looked at their light sensing cells, they had too much actin in them compared to their unaffected relatives. Now, actin is the skeleton, the protein that forms the skeleton within your cell, and it's involved in lots of things like cell migration and trafficking of proteins within the cells, but it's also very, very important in regulating cell shape and the membrane curvature in a cell. Actin has long been thought to be involved in this disc formation, in this outer segment turnover, and we've known that for about 40 years, ever since people showed that there was actin at the site of disc formation. And also, if you remove actin, you dysregulate disc formation, you dysregulate this outer segment turnover. So we wondered if RPGR mutations are causing too much actin, is RPGR actually involved in disc formation? So first of all, we needed to show that disc formation was definitely actin mediated, and to do this I went over to the lab of Ted Wenzel in Houston in Texas, who has pioneered the use of a microscopy technique called cryo-electron tomography, which allows you to look right into a cell's body at an ultrastructural level in their native state. And by doing so, we were able to see that there were microfilaments extending from the cilia in a photoreceptor into this new disc, this new outer segment that was forming. And whenever we looked at thousands of these microfilaments and averaged the shape of it and got the average shape, we realized that it was actin. So it is actin, this sort of skeleton within the cell that is pushing the cilia membrane out, which allows you to form this new outer segment. We generated mice which had a mutation in RPGR, and what we saw was that this outer segment, which I'm showing a picture on the left here of a wild type mouse, a healthy mouse, and in this photograph, what I'm showing you is healthy outer segments with hundreds of disc-like processes stacked nicely on top of each other. In the RPGR mutant mouse, this didn't happen, and the outer segment was distorted. The discs were split and spaced out, and the outer segment was shorter. And at the base of the outer segment, the RPGR mutant mouse was shedding lots of vesicles. So clearly, outer segment turnover was abnormal in these mice. And this, of course, would be in keeping with the human data. Whenever you look at the back of someone with RPGR mutations, they don't have an outer segment. Whenever we looked at the RPGR mutant mice, we were able to see that at the level of the connecting cilium, just at the base of the outer segment, I'm showing photos here, in red is the outer segment, and at the base of that, there was far too much actin in the RPGR mutant mouse. So they're both in human stem cell derived photoreceptor cultures and in mice with RPGR mutations, there's too much actin where these outer segments are being formed. So we then looked with cryo-electron tomography in Houston, and again what we saw was that the actin fibres that we could see extending into the newly forming disc, into the newly forming outer segment, they were far too long whenever RPGR was mutated. So this suggests that RPGR is perhaps controlling the length of these actin microfilaments and therefore controlling the rate of disc formation. So those were lovely experiments, but actin turnover, actin is a very dynamic protein, it's constantly turning over, and so we needed to somehow assess actin turnover over time, and so we developed a retinal slice culture technique, and I'm showing here two movies on the presentation, one at the top of a wild type retina and one of a mouse retina with an RPGR mutation. And what we were able to see by developing this slice culture technique is we were able to watch these actin microfilaments at the base of the outer segment over time. And what we could see is that in the wild type mouse, these actin microfilaments move constantly, but in the RPGR mutant mouse, whenever we got a computer to track the movement of these actin microfilaments, they were moving much less frequently whenever RPGR is mutated compared to the wild type mouse. So it looks like whenever you lose RPGR, your ability to turn over your actin is compromised. And that leads to actin fibres being too long at the site of disc formation, and therefore you get abnormal disc formation and you get these disorganized outer segments. So then the question was, could you do anything about this? And so what we did was we injected these the eyes of these mutant mice with a drug which got rid of the actin. And if you remember, I spoke to you earlier that with the RPGR mutant mice, apart from having these abnormal outer segments, the mice were also shedding lots of vesicles at the base of the outer segment. And we thought maybe because they were making lots of abnormal discs, because they weren't healthy, they were shedding them as vesicles instead of adding these to the outer segment to continue the turnover of the outer segment. And whenever we remove the actin by injecting an actin depolymerizer into the eye of these mutant mice, the number of vesicles at the base of the outer segment decreased. So that was very exciting, and it was proof that if you targeted this actin, this excess actin, maybe you could rescue the abnormalities seen whenever you have an RPGR mutation. Now, this actin depolymerizer, this drug we used, is a global actin depolymerizer. It gets rid of all actin and probably would be very, very toxic to the eye. So we therefore tried to identify the specific pathway that was regulating actin. And I won't get into the very boring science, but essentially we found that our experiments showed that perhaps the limb kinase cofilin pathway was the pathway of molecules that were regulating this actin turnover in the outer segments, or rather in the photoreceptor, to make these outer segments. And so we treated our mice with a specific limb kinase inhibitor to try to target this pathway. And what we immediately saw in the wild type mouse was whenever you inhibited this pathway, the outer segment discs in a healthy mouse were far too long. So that was showing that this looked to be the specific pathway that was regulating the actin, which was building these outer segments. And again, whenever we treated our RPGR mutant mice with this specific limb kinase inhibitor, we again saw a reduction in the number of vesicles that were being produced at the bottom of the outer segment. So what we think is happening is we think that outer segment disc formation is an actin-driven process, and we think that you need RPGR to get rid of the actin once the disc has been made. You need RPGR to facilitate the actin to be disassembled, which allows this new disc to be added to the outer segment, and therefore allows this outer segment to be turned over continually and allow for normal vision. And we think that whenever you lose RPGR, you lose the ability to get rid of this actin, and therefore you have far too much actin, the discs are abnormal, they're shed as vesicles, and it results in photoreceptor stress and death, and therefore sight loss. So work continues in this project to see whether we can target this pathway to try to actually rescue the vision. So that's me finishing. Apologies, I've run a few minutes over, but essentially I'm just going to summarize that what I've spoken to you today about is about gene therapy and how we already have a gene therapy for a very rare IRD in clinic. There are several more in clinical trial. There are other approaches that are in clinical trial that may prove viable, including including a treatment for those with extensive retinal degeneration. And finally, I've just spoken a little bit about the work we are doing, where we are hoping that better understanding disease mechanism of these genes may lead to potential therapeutic options being identified. So I'm going to stop talking there. I just want to thank everyone in my lab who carried out a lot of this work. The stem cell work was carried out in the lab of Charles French Constant whenever I was doing my PhD. I very much work, my mini lab is within the lab of Pleasantine Mill, who works at the Institute of Genetics and Cancer. I want to thank the imaging facility and the mass spec facility in our institute. Also, I want to thank Ted Wenzel in Houston, Vadim Arshavsky at Duke University in Carolina, and also Laura Machesky, who was at the Beatson Institute in Glasgow, but who's now in Cambridge, who were all collaborators on that project I presented. And I want to thank my funders, the Wellcome Trust, Fight for Sight, Retinal UK, the Macular Society, and the College of Surgeons of Edinburgh, and of course the patients in NH Lothian who gave their pound of flesh for the stem cell project. And of course, I'd like to thank you all for listening. Thank you very much.
Brilliant, thank you, Roly. There was a lot of science in there, and although I'm, an expert in PCD and RP, a lot of it still goes over my head. But as a patient, or married to patients and got patients in the family, I think it's really exciting. And I got very excited when you said there was optogenetic therapy, because I know, and I put a question in, I think I'm the only person that's put a question, was like, are we still looking for new genes? Because we understood that you needed to know the genes before you could get it fixed. And if you don't know the genes, you've got a bit of a problem. But that might not be the case. So
yeah, no, absolutely. So, with gene therapy, you need to know the gene, and there is still, I would say probably 60 to 70% of our patients have a gene identified, but there's still a huge amount of patients that we don't know their genetic cause, and therefore they will never be amenable for gene therapy unless we can identify them. And like I say, we are trying to improve to find that missing heritability, those patients that don't have a cause identified. So so therefore, can you develop these gene agnostic therapies, these therapies that regardless of your gene, you can get a treatment? And optogenetic therapy is definitely a potential. It's very early days, and the other great thing about that approach is that it could treat patients who have completely lost their sight in theory, because you're targeting these bipolar cells rather than the photoreceptors. But our lab and many other labs around the world are also targeting are trying to identify how cells are dying and the pathway disruptions that are occurring in photoreceptors in RP, regardless of the underlying genetic cause. So we wonder, are there final common pathways that that are disrupted that result in the photoreceptors dying and therefore result in your sight loss? And if that was the case, then hopefully in the future we will be able to identify targets in those pathways to try to stop the light sensing cells degenerating, and those would in theory be applicable regardless of the underlying genetic cause.
brilliant. Thank you. I know that I asked Kerry to ask the questions, but we've only got one, which is from Chloe, who is from Usher Kids. She says, are the plans in the UK to gather natural history data from individuals with Usher syndrome, given how important this data is to access potential treatment effectiveness? And then plus, what is your view on antioxidant therapies such as Nucuity?
So, thanks, Chloe. So the Rush 2A natural history study has just been completed, and with that we now know, I don't know how many centres around the world took part in the Rush 2A study, but it was a lot of patients, and therefore we've got a really good grasp now on the natural history of Usher syndrome caused by mutations in USH2A. There are other Usher's genes, but USH2A we now have a good going appreciation of how it progresses, and therefore we have a good idea of the therapeutic window in which any treatment might result in improvement. And therefore I spoke about the exon skipping therapy, which we hope Thea will get off the ground, by next year at the latest. And they very much, so Rush 2A was presented at Arvo, and they very much will take that data and use it to plan the trial, the trial protocol. So so we do now have good natural history data on USH2A.
Good.
As far as antioxidant therapies go, and sorry, Chloe, I agree. The lack of data for other Ushers types seems a barrier. Yeah, we could be tackling now. Yes, absolutely, and natural history studies for all the IRDs are needed, and some of them are very rare. What 1B isn't a particularly common gene, and therefore it's going to be much more difficult to get good data on those types of genes. USH2A is a common one, and therefore that's been really comprehensively tackled. Antioxidant therapies, I don't prescribe them. I don't think the clinical trial has been done yet, which definitively shows that they do show improvement. I spent some time in Duke University last year, and some of the ophthalmologists there do prescribe them, and I think maybe in the US they're prescribed a little bit, but I don't, because they're not NICE recommended. There's no proper evidence, but hopefully the trials which are taking place at the minute will show that they are improving, but I haven't heard, that data hasn't come out yet, so I'm that is very much a sitting on the fence answer. Hopefully it will work, but I don't know of any proper evidence to show that it does.
Okay, brilliant. Thank you, Roly. If anybody else has got any more questions, then I'm sure you can help answer them on the chat. If you're staying for a bit,
absolutely,
that would be lovely. Thank you very much. Okay, so our next speaker, I think on summary though, that was really very interesting, but it was also, it gives our RP community such great hope, really, and we'll be supporting you all the way, and trying to do whatever we can do to help you get some of these projects off the ground. So, okay, so our next speaker is Steve. Steve is the co-founder of Cure Usher. And it's a charity dedicated to raising awareness and funds for the cure for Usher, curing Usher, basically, and it's the leading cause of genetically inherited sensorineural, never heard of that word before, combining hearing and sight loss. So, like many of us who are involved in the Ciliopathy Alliance, the principal drivers for all of us are much better life for our loved ones affected by these rare diseases. And Steve is married to Jo, who was born with profound hearing loss and later development retinitis pigmentosa. But Steve's had a varied career, from making acoustic guitars and being a paramedic, and now as the chair of Usher. And he's really passionate about improving the lives of people, and has great, unique ideas on how to raise awareness of the condition. I'm sure he's going to tell you about some of them now. So thank you, Steve, for coming today, and we hope that we'll learn a bit more about Usher, but especially for our audience today, why research is so important for these patients. Thank you, and I'll switch myself off and leave you to it.
Certainly, thank you very much, Fiona. I mean, there's a huge amount of imposter syndrome going on in this kitchen at the minute. But yeah, I'll certainly do my best to put my point across. Thank you so much, Roly, for all that you do, and I think that is a good starting point, because I think this is the thing that really drove Joanne and I to found Cure Usher, was that sense of hope, and that there were people out there that were prepared to and are making careers out of striving to find a cure for inherited retinal dystrophies. And in our case, specifically Usher syndrome and the three types of Usher syndrome that there are. We, in 2018, after, well, Joanne living a lifetime up with Usher syndrome and working in the non-profit sector, just became slightly disillusioned, and and was beginning to lose hope, and more, not so much, I suppose, really quite a harsh way of saying it, but it was more of a resignation, having been told on numerous occasions by, I'm afraid, numerous clinicians, that there wasn't really much we could do about this, and well, there are supports, press services in place. Go and live your best life. And that is almost a quote from one of them. And this isn't just an isolated incident. This is across the board from a great number of people that live with Usher syndrome have contacted us and have told us these stories. It's off the back of that we thought it was time to address this, or at least attempt to try and address this. And that's not to say that there weren't services out there and there were charities already doing this, but there was nothing that we felt, and certainly the people that we were talking to at the time felt that it was specific for those with Usher syndrome. It's a strange one. It's almost like as if it's a little bit of a not so much a dirty secret, but we didn't really know where we belonged. There was definitely a sort of, are we over in the hearing loss sector? Are we in the sight loss sector? We're a little bit of both. And as such, nobody really knew what to do with us. That was the sense that we were getting. And so we came across a great number of people that were essentially self-advocates. And this happens across the whole rare disease sector. Of course it does. Everybody does this. And it's exhausting. I could see this in my wife. I could see this in other people that we met. That sense of frustration and resignation was, well, basically we've had enough of that. Let's see if we can address that. And this was sort of trying to keep up in the recent improvements, the uptick in results that have come from the science sector, the people like yourself, Roly, who are smashing it out of the park at the moment with retinal dystrophies. It gives us so much hope that it's almost like the four horsemen of the apocalypse are sort of they're sort of circling this thing that we need, that we would rather not have here. Thank you very much. And it's that hope from your side that we try and match from our side, from the patient perspective. Unfortunately, there seems to have been a lot of time and a lot of investment in the bridge, in that infrastructure, rather than focusing on the people that are actually crossing that bridge. It's almost like as if there is a thing there to look for and to tick a box, and we are involving these people, but you're not talking to these people. And it's not about apportioning blame. This is a very dynamic and it's a changing landscape at the moment, and it's changing for the better, which is great. But there's a chance that we may slip into tokenism, whereas we are simply ticking boxes, and have we talked to the patients? Have we consulted the people that are at the end point of this process? And it's great to see in all the conferences that I've recently attended, and previous to this, of course, that this is starting to become an issue that people are recognizing. And it's even from the NIHR survey in 2018, the I Am research, they identified that there was a willingness there. Of course there was. These people with a rare disease, with any sort of rare disease, were willing, more than willing, to engage in clinical research. But where was the opportunity? And there's a slight disconnect between these wonderful people that do work that is so far and away, the science behind it is astonishing. I mean, you know, these are I often quote this throughout my career in the NHS. These people forget more than I'll ever know. These people is terrible, isn't it? Even I'm doing it subconsciously. There is a bias that you are so far and away these amazing people that do this work that we've lost that sort of bridge between us, and we've trying at Cure Usher and with other patient advocacy groups such as Usher Kids, Retinal UK, Fight for Sight. We're all there to try and talk, to try and bridge this gap, to try and get our point across. And I think that's what one of the first things that we wanted to address with Cure Usher was that awareness, because it's so very easy. I kind of feel like I'm talking in a room because you probably all know this, but it's so very easy. I think it's worth saying it's so very easy to to talk to each other about how terrible it is, and to try and raise awareness within, you know, our own environments. And yet we leave a conference, we even leave our front doors and go to the co-op, go to we get our train. Nobody knows what it is. Joanne walks with a cane, a white cane with red stripes, denoting that she has a hearing loss as well as a sight loss. Nobody knows what that is. It's in the highway code. It's there, but it's just about making people aware of it. And it's not about apportioning blame. This is it's about a sort of a sea change in how we look at things. If you think of the Apple shops, you're a massive multinational, multi-international company. When you go into an Apple shop, it's a totally different experience, or it has been. It was one of the very first shops that would just seem different. And it was almost like as if the people that were in that shop were not the end users. It was the people that actually engaged with that shop. But it was a clever trick because you sort of thought that these were amazing people. This is just it was that sea change it was so cool. And still is in many ways. It was seen to be so cool to have an Apple phone, and it was a very clever trick. There's a lot to be learned from the people in retail in convincing that this is the right way to do it. And I think that patient engagement it should be central to it. The recruitment of patients to clinical trials should almost be a byproduct of that patient engagement. You should put the humanity first, the people at the forefront of this. And by that, they just become part of the process of research. So people like Fortria and Avantgarde, who are doing they're setting on a new process with some Italian scientists for Usher 1B. They recently consulted us. So you know this is happening. This actually is. You know, we're starting to see the results of starting to bang our drum, which is it's been needed for a long time, I think. But the hope is there. The research is there. Things are happening across the globe. There are numerous research things underway, and it's fabulous. It's a strategic approach, though. It's about awareness. It's about designing the right kind of research projects, recruiting them. How do you recruit these people? You've got to contact your GP. It all starts with the GP, and unfortunately, most of our GPs in this country are massively overrun. They're overworked. But this is about you as a patient to be able to give them a bit of a helping hand. I always used to say, as a paramedic, if you can't, now I've attended you in an emergent situation, you need now to go and tell your GP and build up a casework. And this is what we need to do in the rare disease environment. Take a step back, tell your GP, get them to refer to your specialist centre, wherever that might be. There are so many now that Rare Disease Research UK have now established some 4 hubs across the country. It's starting. This sea change is starting to happen, and we're keen to try and put Usher syndrome at the forefront of that. it's not as if that there are uh, there are it's a larger cohort than you might think. There are potentially 11,000 people across the country. It's the same people that have cystic fibrosis, same number of people. It's we're like Margot Ledbetter said, we are the significant minority. It's worth backing. And I think if we can hold our nerve, if we can hold that hope and keep humanity at the center of it, then I think we've got a good case in going forward. And I think that's where I think I've said enough. I think I'll invite you for any questions, but yeah.
I thought you were going to tell us about how your fantastic awareness of how you have put awareness on the forefront of everybody's sofa on 5 nights a week. So I think that's quite an important story.
I think
we're all very jealous of that.
I think it's part of using what you've got in your arsenal. What part of what you have to hand is you've just got to do what you have to do. That's what we held close to our hearts when we started this. It was just having the nerve to do it. It was, and the people that you meet along the way, it's surprising what they have to hand. It's surprising that even the people that you might not think you have anything to offer. I'm sat here, my heart's in my mouth. I absolutely hate Zoom calls. It's dreadful, but I'm trying my best. You've got to do it. I think of all those people, not just of Joanne, but of all those other people that are connected with it. There are thousands of people behind this wall that are connected to Usher syndrome. And in keeping that in mind, you've got to kind of look for what you look for the people that you can depend on to spread the word. And luckily, everybody's got to have a slice of luck, of course. But our patrons were lucky enough to secure some work, and yeah, to get a storyline of Usher syndrome in a national soap was to persuade them to engage with that was huge, absolutely huge. And just to be able to drop that into everybody's, like you say, everybody's tea time, just whatever it was, but it's starting a conversation, and that's so very important.
So I think quite a lot of us will relate to the when you said, oh, you go home and just live your best life. I think we were told exactly the same when Stuart was diagnosed with RP as well.
I think this is it. It's everybody is in the same boat. That's what I say at the start. I feel a great sense of imposter syndrome, insofar as you all know what this is like, and you're all behind me. I'm all behind your own ventures. We've all got to fight for this. It's massively important. And it's not about apportioning blame. It's about pushing this forward. Everybody wants to help. Everybody that I've come across, as not one of them has said, no, I'm not having anything to do with that. Not at all. It's just a bit, this is a very dynamic scene at the moment. Things are changing so fast, with developments not only on the science side, but in funding the research, in convincing people that this is worth backing. If you can solve the problem at the start, you don't have to provide the stuff to sweep it up afterward. If we can cut that corner, then isn't that what we all want?
Yeah, I agree. I think we're really pleased that you, Cure Usher, are part of the Ciliopathy Alliance now, and and hopefully we can together
And I think that's a point in itself. I think we were signposted to you by Chloe Joyner from Usher Kids.
Yeah.
There's a beautiful example of cooperation, of going, I know that person, that you need to talk to this lot. That's what we want. It's no good having these things, it's, I mean, you'll have come across this, Roly. in trying to get your name on a paper, a research paper, you want to be the first on that paper, and then there's all these egos bouncing around, well, I did this first and all the rest. Great. But really, in 6 months' time, nobody's going to give a hoot about that. This is about, you're doing this for the long term. And it's also about it's also about the fact that we live with this 24/7. We have people, I've anecdotal evidence, people's stories that they've told to us of, people coming in for assessments. And then when you shut the door and they leave and they leave this behind. Whereas, we're the ones sitting in bed at 2:30 in the morning with our toes curling and go, oh God, what do I do about that? It's not our job to provide evidence for you. It's not an us and them. We live this. The design of research should be around the patient, and it's not us to fit around you. It's easier said than done, but like I say, this is a dynamic environment now. Things are on the upswing, especially with retinal dystrophies across the world. It's all of a sudden, it's gone. It's an exponential curve, which is lovely. It's great to see that it's not linear. I haven't got time for that. We need these things. But this is what happens. You learn on, and it builds on that. It's so encouraging to see.
I think, okay, if I can just barge in, I think just what you touched on about patients being involved in research. I think the funding bodies as well as the charities are increasingly recognizing the need for this, the importance of it, and the requirements for it. And so, all grant applications now have quite a big section on how you've involved patients in and relatives in the design of the study, how and also how questions about whether patients and relatives are involved in carrying out the work or analyzing it. There's even they even want that is obviously seen as beneficial and so the funding bodies are realizing that it's research will probably be better with a strong patient voice.
That's it. Yeah.
And you also mentioned about first author arguments, etc. There's no doubt collaborative science is so much more fruitful than
Huge.
pitching people against each other.
Exactly.
So I don't have anything really other to say about that other than I agree. And the smaller your ego and the bigger your research network, the more success you have, without a doubt.
Exactly. Because it's almost like the argument against plagiarism, isn't it? Plagiarism is just taking one thing from somebody and using that. Whereas if you do it enough times, it then becomes research. It's a strange argument, and it's a small enough room to begin with. The rare disease space, and then it's small. It gets even smaller. It's in the sight loss sector. It gets even smaller. It's retinal dystrophies. Crikey, we in the bigger scheme of things, we can't afford to be wasting time, grandiosing and showboating about various things. This has to be an approach that we're all together with. Yeah. And retention and follow up from research is huge. What are the things that patients want out of it, and what do they see as the benefits from it? Obviously the cure, but going around that and building up a sense of purpose behind it as well and around it, as well as the end point. It's like Maslow's triangle. It's got to be, it's built on, it's a structural approach. It's got to have the right places, the right pieces in place, not just the very top that everybody, quite rightly so, strives for. But it's a strategic approach, and if you're aware of that, and again, this is I've just said it. It's all about awareness from the bottom.
Helen would like to say something actually about collaboration, and I think she'd probably say something about the patient support day in Cilia 24 as well. I think
No, well, no,
No?
obviously I can, but first of
You
all, Steve,
can.
I really, really appreciated your talk. I thought it was really, really, really great and really motivational and inspiring. And I know how awful Zoom talks are. So I think it's nice that you also just mentioned that, because I think, it's kind of the elephant in the room with these Zoom calls. But I also just kind of want to say something that I think obviously having watched this cilia field grow for the last 20 years, because it's scary to think that I really have been here for 20 years. And it really, it's not linear, you're absolutely right, and just how the amount of new information and kind of ideas that are going and kind of development of treatments, really there's a lot going on behind the scenes as well, and it absolutely is not linear. Absolutely. But I also think that a part of the reason why the ciliopathy field that's happening is the association into the action and the connection that we have to patients, more so than in some of the other fields. And I obviously there's a long way to go. That's kind of the reason why we have all these patient days. We're bringing the patients into the scientific meeting, but I really do think that it really is. Of course, it needs to grow, but already we have only come so far because of the close connection that we have with the patient.
Absolutely.
And there's a lot of medical researchers out there who've never, ever didn't even know,
huge amount
contact to patients.
Huge amount. I made a note before, 1 in 20 cancer patients enroll. It's only 1 in 20. I mean, and that's cancer. And this is the thing. There's some great research done by Fight for Sight and Time to Focus. I recommend you read that, whatever your background is. And they would say, that the investment in sight loss is only 1.5% of the whole of central government funding for genetic research. 1.5% of it was put towards sight loss. So the lion's share of it went to neurology, respiratory, cardiology. It's they're the big players. So we've got to use what we can to bolster our position. And I think the biggest thing we have is the fact that we've identified patient engagement, amongst the first ones to do it.
Yeah.
We recognise that importance.
Yeah, £9.60 per patient per year.
It's mad, isn't it?
It's awful. And you spoke about earlier about funding and on how we are being recognized more, rare diseases being recognized, but sight loss is still so underfunded, as you say, despite the fact that that time to what was the fight for
time to focus?
Time to focus. It showed that sight loss is much more impactful than rheumatoid arthritis, than some forms of cancer. The impact on the patient, So yeah, we are underfunded, and we need to think cleverly about how best to use the money we have, and the money that is hard earned by the charities, or hard raised, should I say, and formation of networks, research networks, is going to be crucial if we're to spend that money wisely.
Hugely important. There is a symposium June the 6th in Oxford, the Harwell Centre, on that very subject, on rare disease research funding. It's even there, it's a massive subject, and it's like you say, that the sight loss sector. It's almost like it's a story I tell over. I'm going to tell it. I'm sorry, but it's an unconscious bias. Every textbook I ever came across when I was doing my training as a paramedic, it was always the sight was always at the back of the book. How are you even reading the book? You're using your eyes. It's just ridiculous. It's almost like I said at the start. It's almost like it's like a dirty secret. But your eyes. Oh, I don't want to do that. Oh, there's a cringe about it, and that needs overcoming. It's because it's not. It's it's a hugely important factor, and the fear behind it that can be broken down by education, by awareness. And that's, speaking to people such as yourself, Roly, and to everybody in this room, is it's so encouraging. And it emboldens you to speak up as well, which is so important.
Yeah, here.
So, have we got any more questions? I don't think we have got any more questions, have we? Have you got a question, Kerry?
Yeah, just to add to that, I think it's one of the areas I find really frustrating. I remember when my son got a diagnosis, we were told it could be 10 years before, that there'll be any type of gene therapy or anything like that. 23 years later, we're still saying the exact same thing to people who are having children diagnosed with the condition. And I think even with some of these horrendous conditions, Alström being one of them, it can affect the heart, diabetes, liver failure, kidney failure, some real, life-threatening things. But actually the number one research priority amongst our families is sight loss, and wanting to do something. And I think it's, as a patient organisation, I find it really frustrating because it's, you've got so much enthusiasm and families wanting to do crowdfunding and things, wanting to raise money, but it's so difficult when you don't know what it is that you're raising money for, or where you want to put your efforts, or, because you need that. You need somebody with a real interest. And even, we are so lucky to have a highly specialised service, and, but we've still not really got anybody that's interested in doing research into sight loss. And so where do you get them people from? How can you? And I think, we're just a small condition, ultra rare condition, but I think I suppose always trying to be hopeful that all of this research that's being done in some of these areas for some of these sight loss conditions will ultimately benefit conditions like Alström, like BBS and others. But I sound like I'm really moaning, aren't I? But I think it is that frustration that I think a lot of us feel.
Definitely.
I think as far as gene therapy goes, as far as novel therapeutics goes, I think you talked about the 10 years and the 23 years later. I think it took a long time for Luxturna to get over the line, because of red tape, because of for a good reason. But now that we know that gene therapy in the eye is safe, I think there will be an acceleration of trials, and therefore products coming to market, because I think you might even be able to do away with the animal model because we know it's safe, and maybe a cell showing that you can rescue it in a petri dish is enough. I think that we will see a speeding up of therapies coming to market now. ARVO, the big American meeting I was at last month that I alluded to, it was awash with safety trial data for retinal dystrophies. I cherry picked a couple, but there were so many talks. It really is. There is a growing there's a groundswell.
Definitely. I think that's what's so encouraging, isn't it? That there's that recent uptick, that upswing in results. And like you say, that you build on what your previous work. And like you said, we've had some inquiries recently about people feel uncomfortable about cutting edge research involving animals, and that it's a fair question, it's a fair point. And like you say, the more that we do, we can accelerate past that. If you can prove that this is safe, that you can prove this is safe in that environment, then you don't need to do that part of it. But again, it's all about it's awareness, it's education, letting everybody know about it, and the more that you talk about it, the more the chance you have of finding people that are interested in this, in getting past that uncomfortable feeling about it, and to want people to invest in sight loss research.
Brilliant. Okay, unless anybody's got any other questions, I think I'll just say thank you to both of you for a very enlightening afternoon again. Lots of stuff flying through my little brain here, trying to work it all out. I probably might have to send you an email later, Roly, to get you to explain some of those terms. I hope everybody's enjoyed it. I thought it was absolutely fascinating, but I mean, I just think we need to. I think the key things for me is we need to make sure the patients still have a voice. So if you're a patient here, ask your doctors about research, tell them you want to be involved in research, tell them you want to do anything you can to help. If you're a researcher, make sure that you're in contact with your patient groups. We're a a bossy, interested, keen to help group. So yeah, keep asking us for your help, and I hope that you'll join us for the next one, which I think is in about 3 months' time. And if you have attended, you've got anything specific that you'd like us to talk about, then please, feel free to contact us through the website. And most of you know me anyway, or directly email me. So thank you very much to everybody for coming, and thanks again to our speakers. Really, really, really great afternoon, and thank you, Tonya and Kerry, for your helping us with the admin. And we'll see you all soon. All right, take care. Thank you.
Thank you.
Bye.
Thanks a million.
Bye now.
Bye-bye.
Bye.